it's really hot outside, but you took a walk, maybe 15 minutes to walk to attend this wonderful seminar. So I really appreciate it. So it's my great pleasure to introduce today's speaker. His name is Taekjip Ha. People call the TJ Ha. So currently he's a professor of the Bloomberg, the distinguished professor at biophysics and biophysical chemistry and biophysics and biomedical engineering at Johns Hopkins University. So he is the Howard Huey Medical Institute investigator. He graduated from the Seoul National University, which I graduated from, and then he moved to the Berkeley, and he received a master degree, and he received a PhD degree there. And he worked with the famous Simon Weiss and Daniel Chemela, and then he worked with the Nobel Prize laureate Stephen Chu. Then he published a numerous paper, and then he starts his career University of Illinois Urbana-Champaign. Actually, he was my thesis committee member, actually. So he asked a lot of interesting questions, sometimes difficult questions. Anyway, so he was promoted associate professor, and he was promoted full professor. And then recently he moved to the Johns Hopkins University. And he is actively working on the single molecule biophysics. So he received a lot of awards, and I will introduce some awards. The most, I would say, honorable thing is he is a member of the National Academy of Science and the American Academy of Art and Science. Also, he is the prestigious Hoam prize in the science, the Hoam Foundation. Actually, this is related to Samsung, and he has a lot of the award actually. So, and yeah, without further ado, let's welcome the Professor Ha.
So yes, I was on his thesis committee. I don't remember asking him difficult questions. So can you hear me in the back? Okay. So I'm really pleased to be here. My first time to Hong Kong. I didn't realize that Hong Kong has so much nature. I had a great time walking around the mountains and the beach, and great food in Sai Kung already twice. And I look forward to enjoying more of Hong Kong today. So I wonder how many of you are physical scientists? Okay, biological scientists. So I'm a physicist by training, but I tell people that I forgot most of my physics, but I haven't learned enough biology to call myself a biologist. So to paraphrase Britney Spears, who sang, "I'm not a girl, not yet a woman." So I feel like I'm not a physicist, but not yet a biologist. And so when I give a talk, in a biology audience, I tell them I'm a physicist. And in a physics audience, I tell them I am a biologist, so no one asks me difficult questions. I'm not sure what to call myself in this audience. Let me do my best. So we do single molecule measurements. I call them nanomachines because they are very small, just a few nanometers across. And they are really like machines. As I learn more and more about these molecules, structural biology and their function, they're amazingly tailored to perform their functions in a precisely tuned manner. Today I will talk about a particular nanomachine called helicases. Let me introduce what they are, and then tell you about the technology we developed to study these proteins, and then show you some interesting data. And finally, some of the biotech applications we are pursuing based on one of our discoveries. This paper was published more than 60 years ago. They proposed a double helical structure of the DNA, and then they ended that paper with this paragraph saying that it has not escaped unnoticed that the specific pairing suggests a possible copying mechanism for the genetic material. This was such a modest statement. Many people consider this an understatement of the century, the previous century. And in fact, the authors worry that someone else may put that statement in a more explicit manner and take the credit for it, for the discovery, along with the big prize, the Nobel Prize. So they published another paper in the following months in Nature, proposing base pairing between A and T and C and G as a mechanism, and the rest became history along with the Nobel Prize. This paper then ended with this paragraph. Okay, even if we are correct, there's much remain to be discovered. For example, if the DNA chain is tightly built, then how do you separate and unwind a pair of chains when you must? For example, when you need to copy the DNA. It took another 25 years until people discovered a class of enzymes called helicases. What do the helicases do? So this is my helicase, and the helicase is supposed to run along the DNA as a motor protein, just like a car moving on the highway. They're using gasoline as fuel. A helicase can move on the DNA using ATP as a fuel to cause unwinding of the DNA into single strands. But if you give a helicase protein a single-stranded DNA, then it can also move on the DNA in a directional manner using ATP, most likely one ATP per one base of DNA translocated. And single-strand DNA has a polarity. One end is called 5', the other is called 3'. So some enzymes move from the 3' to 5' and vice versa. We care a lot about the helicases because they're important, for example, for genome maintenance. Anything that handles DNA or RNA actually has helicases functioning in the process. And they can do unwinding of DNA or RNA. They can remove proteins bound to DNA or RNA. They can cause many other reactions. And if they are not properly functioning, if you have mutations in some of the human helicase genes, you can develop serious genomic diseases, genetic diseases such as Bloom syndrome and so on, and some causing a high incidence of cancer. So their proper functioning is very important for survival. And they are classified into several classes. Some helicases form a hexameric ring, and others do not. And some work on DNA, and others work on RNA. And some have this 5' to 3' polarity, or vice versa. So what I mean by this is following. For 3' to 5' helicase requires 3' single-strand DNA tail to first load onto the DNA, and then it moves in the 3' to 5' direction to displace the top strand. 5' to 3' would work in the opposite direction. The enzymes that I'm going to tell you about are non-hexameric DNA helicases that unwinds DNA 3' to 5' direction. Okay, we use a technology called single molecule FRET, fluorescence resonance energy transfer. If you want to measure the conformation of a single protein, you can put two dye molecules on the known site of the protein, green and red on these locations, so that you can distinguish between the open and closed conformation. In the open conformation, you excite the green dye using your laser, you get green photons out. But in the closed conformation, within a few nanometers, then of separation between the two dye molecules, then energy can be transferred directly from the green to the red molecule, red dye, without any real photon emission, and you get red photons instead. So by measuring the relative intensities of green and red, you can deduce the conformation, whether it's closed or open. Now, if you want to measure how quickly butterfly flaps its wings in ensemble, you must synchronize the reaction, for example, by convincing them to start from one conformation sitting on a leaf. Then you can frighten them into taking off the leaf and perform a relaxation analysis. However, as you can imagine, this does not necessarily give you information on wing flapping, and this may reflect mainly how long it takes for the butterflies to take off the leaf. Okay? So ideally, you want to look at a single enzyme as a function of time, and then measure anti-correlated changes in red and green intensities. That is your signal. And you can do these measurements routinely in many, many laboratories. So my first PhD paper was actually to demonstrate that this can work. And thereby you can deduce the time scale of the fluctuations and the amplitude of the fluctuations. There's another powerful single molecule technology called optical tweezers. I call these chopsticks of made of light, as shown here. And the idea is that you use a focused laser beam to grab a small microsphere or bead in the middle of the laser trap. And then you can play a tug of war between the enzyme sitting on the surface, pulling on the DNA, and you can measure and apply forces down to piconewtons, and also measure the movements of this enzyme on the DNA, sometimes down to a few angstrom resolution. A really, really powerful technology. So many labs have tried to combine single molecule fluorescence or FRET using optical tweezers. So there are many examples, and I'm going to tell you our own version of the instrumentation. So the goal is to measure conformational changes of molecules using fluorescence, FRET, as a function of applied force. Why? When you do optical tweezers measurements, you're performing a purely mechanical measurement, but with your eyes closed, like that. Whereas if you do fluorescence measurement, that as we do normally in my lab, also in Hyungun's lab, you're making passive observations with your eyes wide open, but with your hands tied in the back, like that. But by combining the two, you can hope to sample the best of both worlds. So many years ago, my former postdoc, Sung-Chul Hong, developed an instrument and used it to measure DNA conformational dynamics as a function of force. Then a student here, Rubo Zhu, became more ambitious. So he said that now I want to measure DNA-protein interactions. So here, single-strand DNA is wrapped around a protein core, as shown. And then he could grab the bead and then apply force to the end of the DNA so that as the DNA is unraveled from the protein core, he could measure decrease in breadth. This is actual data where we increase the force as a function of time, the gray curve, from low value to high value. And then we come down to an original value, repeat it 5 times, and measure the red and green intensities at the same time. As you increase the force, green is going down, green is going up, red is going down, because you are increasing the distance and decreasing the proximity. And then you can also plot what's called the FRET efficiency as a measure of proximity, which is high at the beginning and goes down gradually. You can really show that with force, you can unravel the DNA little by little from the protein core. So when Rubo showed me the data for the first time, I was so impressed. Data was so beautiful. So I almost cried. But I didn't cry. Like every good advisor, I told him, okay, that's nice, but go back and take some more data. Okay. Anyway, so that was a really fun experiment. And then Robo trained another student. He trained a new Robo is now actually a postdoc with Xiaowei Zhuang at Harvard. And then he trained my Vietnamese student, who is now a postdoc with Stephen Quick at Stanford. And then she became even more ambitious. She decided to unravel now double-stranded DNA around a protein called histone core. This is what's called a nucleosome. And then she was able to also measure Fred decrease as she increased the force around this protein structure, DNA protein structure. And the surprise was that when she pulled on it and then measured Fred on one end of the nucleosome, unraveling occurred at a low force, whereas when she pulled on the other end of the nucleosome, it unraveled at a much higher force, showing that asymmetric unraveling is happening here. And based on this, we also made a very interesting story. Then we got even more ambitious. So Jan Shemna was a colleague of mine in Illinois, who is an expert in high-resolution optical tweezers. So we hired a postdoc together, who is a physicist, Matt Comstock, to build ultra-high-resolution optical trap with single angstroms resolution, but that can also detect single molecule fluorescence. And there we had to have two laser traps trapping two beads and DNA in between, and then laser confocal laser detection in the middle. Let me see if the movie works. Because if you have the trapping laser on at the same time as a fluorescence excitation laser, photo bleaching of the dye in the middle is 10 times faster. We had to actually interlace the excitations, going back and forth, and actually we had to do this 60,000 times a second. So it's pretty impressive engineering that Matt did here. And I'm going to show you data obtained using this instrument later. All right, so coming back to the helicases, we are studying a class of helicases from bacteria. They have different names, RAB, VVLD, PCLA. But you don't really need to care about the names. If you do care, you can look at the bottom here to see which helicase I'm talking about, but I'm just going to call them helicase. Then they are 3 to 5' DNA helicases, and structurally similar. They function in important aspects in bacteria. And they undergo a large conformational change, as seen in this animation between two structural states. And the questions we can address are, what are the functional forms for DNA unwinding? Is it going to be a monomer of this protein, or do you need to have two? And when you have open and closed conformations, which of the two is important for DNA unwinding? Finally, this is a really key question: how is unwinding regulated? So you don't want these enzymes to unwind every DNA they see inside a cell, because that can wreak havoc, and you want to regulate the activities tightly. How do you achieve that? You want to make sure that you stop the activity until you need the activity and to tell them to go. Okay? Go or stop. So based on several ensemble-based and single molecule-based studies, my collaborator, Tim Lohman, and we proposed this model for one of the enzymes. So the idea is that you have a motor protein as a monomer that moves on the DNA, the 3' to 5' direction. But once it hits the duplex DNA, it cannot unwind the DNA as a monomer. So it gets stuck there. And then, but if there's another protein coming, then magic happens. You see unwinding. So I call this a monster truck mechanism. But there is a competing model based on structural studies. For example, this is a very young structure of one of these enzymes bound to double-strand DNA with a 3' tail, which is a substrate for unwinding, and with various ATP bound and states. And they proposed that a single protein can unwind the DNA successfully, meaning that once it binds, it can unwind multiple base pairs before falling off. So we decided to test these two models in the most direct way. So here's the experiment. So my former student, Kyung-Suk Lee, who is now a postdoc at Harvard University, he built an optical trap setup, but combined with what's called a turf for measuring single molecule fluorescence near the surface with high sensitivity. So in this experiment, he made a long single-strand DNA tether, thousands of bases, one end on the surface, the other end on the bead, so you can stretch the DNA sideways, then image proteins moving on the DNA using single molecule fluorescence detection, taking movies. So the way we generate the single-strand DNA is shown here. So you have a long double-strand DNA. We use an enzyme that can digest the DNA from one end, and one strand only, like down here. You can either go all the way down, or you can stop in the middle to leave a double-strand DNA that can be unwound by the enzyme. So in this case, because the remaining strand has a 3' end here, if the enzyme binds somewhere here, it'll move down closer to the surface as a 3' to 5' helicase. So I'm going to show you a movie. So that's the surface tethering point. This is where the trap bead is, and DNA, protein will bind somewhere here, and it will come down. There's a junk spot that you shouldn't pay attention to. Let me run the movie here. So something binds here, and then it comes down as a function of time. And that's actually consistent with the expectation that this should bind and then come down the 3' to 5' direction. You may have noticed that it also becomes brighter and brighter as a function of time. Why is it collecting cosmic dust? No. What's going on is that as it comes close to the surface, excitation laser becomes stronger and stronger. That's why you see increase in intensity. And it's actually that you can use that to measure the penetration depths of the excitation profile. If you take a line cut here and plot it as a function of time, it's called the chymograph. And you can show that, yeah, indeed it binds somewhere up here. As time goes on, it comes down and getting brighter and brighter with an amazingly uniform slope. Sometimes we get lucky. We can see many proteins binding and translocating on the same DNA. And again, they all seem very parallel to each other. And you can get the speed from these curves, about 200 bases per second, which is in agreement with the number that was determined by my collaborator, Tim Lohman, using other approaches. So I call this my meteor shower because it's so beautiful. Sometimes we see data that look like this. So this guy on this DNA, it came down a little bit and then it stopped there. And then there's another one that comes down and then it stopped there. Whenever we see two or more events like this on the same DNA, they stop at the same location. So our interpretation is that helicase comes down, and then when it hits a duplex, it cannot unwind the DNA, it just stops there. To test this idea further, we measured the stored position from the image here because we know where the tethering point is. You can measure that distance. And you can pull on the DNA and then measure the length of the duplex mechanically, because duplex DNA and single-strand DNA have different mechanical properties. And you can show that indeed we see a slope of 1, y equal x, supporting our model. We can also do another experiment, which we did based on Vivio's suggestion. We can actually put a DNA at a known position which will present a blockade against translocation. Then you can image the DNA. The DNA is right here. And then helicase comes down and then it stores right where the DNA is. And same here, there was DNA here, helicase comes down and stops at the same position. So if you measure the position of the DNA and then where the helicase stores, they agree with each other within 20 nanometer, which is more or less a precision. So that supports that, the idea that UVLD cannot unwind the DNA as a monomer. Sometimes you do see unwinding. Here's an example where helicase comes down. It stores likely because you have duplex DNA as a blockade. And then later on, it starts to move again. But the slope is different. Here is 200 per second, and here is about 50 per second. It's known from Tim Lohmann studies that unwinding is about 3 or 4 times slower than translocation. So we think that this is unwinding of the DNA. But you may notice that when before unwinding starts, fluorescence signal actually increases. So what's going on is that protein comes down, stores here, then another protein shows up to double the intensity, and then only then helicase can unwind the DNA. So that's the model we have, and you can also use the mechanical signal to deduce that unwinding also starts at the same time point. So I think it's a pretty good evidence that you need actually more than one protein to unwind the DNA in a detectable manner for this enzyme. All right. Another thing that I want to discuss is conformations of the helicases. There are two structures, open and closed, and there are 4 subdomains that are colored. And the main difference between the two is that this blue subdomain called 2B domain is rotated around the vertical axis by about 130 degrees. That's a really large conformational change. You can imagine this domain is actually rotated like that. Okay? And some distances between 2 sides on the protein can change by more than 50 angstroms. But this closed form was crystallized together with a DNA with a 3' tail. The structural biologists actually proposed that this is the functional for unwinding, which is very reasonable. Yeah, so that's the model that I just described. But we also had I had a dissenting view that suggesting that actually the closed form is inhibited off state, and for several reasons that I will skip here. So, anyways, I had an argument with a structural biologist friend, which is active, and so we bet money. We bet 50 cents. So then I came back and told my students, Sinan Aslan, why don't you actually test this directly? Resolve the controversy. So what he did was to engineer 2 cysteine residues, shown in red, into the protein. And if you do it in such a manner that they are very close to each other, 7 angstroms from each other in the closed form, but 40 angstroms apart from each other in the open form, and when you use a bifunctional cross-linker to cross-link the 2 cysteines, then you will stabilize the closed conformation and disallow the open conformation. And we call this rep X because actually it was rep helicase and X for cross-linking. You can also put the cysteines into different positions so that they are very close to each other in the open form, but 30 angstroms apart in the closed form, so that you can stabilize the open form. So this is a control construct, and we call this rep Y. Why? Because Y follows X. So the prediction based on my thinking was that this closed form is inactive, especially no-go signal, and then open form is a go signal. Then my student came back with the data, actually in ensemble measurement, and showed just the opposite. It showed that this becomes actually much more active in the ensemble experiment. So, okay, if that's true, then is it because it forms these dimers better, or is it because now it's active as a monomer? So he performed a single molecule Fred experiment where he immobilized the protein, this cross-linked protein on a surface using an antibody. And then he added DNA, shown here, and labeled with the green and red dye molecules, so that initially you get low Fred due to large separation. But then if you measure this using the wild type helicase, then as a function of time, you can see appearance of fluorescence signal when the DNA binds to the surface immobilized protein, and then the Fred remains low, largely green signal, until its dissociation, and then you see three additional binding events, but no signal change upon each binding event. Same for rep Y, our control construct. Or if you do the same with rep X, here's what you see. You see initial binding in low Fred state. This is Fred efficiency, and then Fred increases, and then decreases to 0, and then the signal disappears, and this repeats 3 times. So what's going on here is that as the helicase unwinds the DNA as a monomer, then the unwound strand becomes coiled up to bring this red dye closer to the green dye, giving you this nice Fred increase. And when unwinding is complete, the red strand is released from the surface, and that's why you see a big drop in Fred to 0. And then finally, when this strand is released, you get loss of fluorescence, and you can restart the process over. So this shows that rep X DNA as a monomer. So by this point, I had to abandon my model. So we revised the model shown here. Right? So now this is the go signal. This is the no-go signal. That's good. It means that my student didn't fabricate the data. He knew what I wanted, but he gave me just the opposite of what I expected. Then how good is this helicase as a monomer? So we then collaborated with Yam Chenra in my previous institution to use optical tweezers to measure activity of rep X, the cross-linked version. So you can tether that protein on a bead surface using an antibody, and you have a 6,000 base pair long DNA, the 3' tail. So you can form a tether, have ATP, and start the reaction. If helicase unwinds the DNA, then this DNA tether will become shorter and shorter. So distance between the two beads will decrease as a function of time. And here is the result. So we form a tether where unwinding is inhibited, and then in the microfluidic device, and then this is a yellow region in time, and then we move the tether to the region where you have ATP. Then as soon as you do that, then DNA length goes down, and it goes down all the way basically until the two beads collide with each other. So somehow rep X becomes a highly processive helicase. They can unwind thousands of base pairs without falling off. And so that was, I was really impressed because there's no other example of this kind. And it is only true for rep X, not for the other two. And we can also show that even when you have very large forces, 20, 30, 40 picojoules, this protein can keep going, and its average speed does not decrease. So we decided to call this a super helicase. And we because it's super. And actually, in our science paper we published, I put a super dash helicase into the title, and then they said that actually there should be no hyphen between the two. So we ended up creating a new word like Superman. There's no hyphen there, right? The next question is, if the closed form is active in unwinding, why did nature create the open form? And so it must be for regulation. So then we turn to this ultra-high resolution optical trap combined with single molecule fluorescence to measure unwinding of a DNA hairpin using the optical tweezers and measure protein conformations using single molecule thread. So here's the hairpin assay. So you have a DNA hairpin in the middle, and then you have this small single-strand DNA region in the 3' side of the hairpin, so that the helicase can be loaded here. And then as it unwinds the hairpin duplex DNA, then distance between the two beads will increase in a stepwise manner. And you can measure using the optical trap. And you can use a green laser light to excite the fluorescence signal to measure fluorescence at the same time simultaneously. And if you can count the number of fluorescent molecules, you can count how many copies of the protein is there. Or you can have doubly labeled proteins for thread measurements to measure conformations. This is not an easy experiment, but Matt Comstock managed to get quite a lot of data. So let me show you one example here. So here in this case, he used a helicase enzyme labeled at 2 locations with a green and red dye molecule. And so that in the closed conformations, they are close to each other, so you get many red emission high thread. In the open conformation, they are far away from each other, you get many green emission low thread. First, let me show you, as a function of time, number of base pairs unwound for the DNA. As you can see, when DNA is under tension, you do see unwinding of few base pairs, up to maybe 15 base pairs or so. So you see DNA is being unwound, and then DNA is being rezipped, unwound, rezipped, unwound, rezipped, and so on. So basically, the helicase protein is sitting there, unwinding the DNA a little bit, and coming back, a little bit, and coming back. Actually, this coming back phase is also gradual, and is dependent on the concentration of ATP. At the same time, we measure a fluorescence signal of green and red channels. And this is the intensity of green and red channels. It's a little noisy because we can only collect data from a third of the cycle. I showed you that you have to go back and forth actually between different laser lights. Nevertheless, if you look at the thread efficiency calculated as a ratio between the red and the total intensities, you see a nice signature of fluctuations, and you can begin to assign that low thread state to the open conformations and high thread state to the closed conformation. Now, it becomes really interesting when you correlate this with the optical tweezers data taken at the same time. So you can color the high thread states in red, these red regions. We can color the low thread state in green, and then look at what happens to the optical trap signal. So when it's red, DNA tends to be unwound, like that. When it's green, DNA tends to be rezipped. So so it's actually it's a little bit more complicated than our previous model, which was already revised. You can do this for many molecules and plot the 2D histograms of the thread value and the velocity of the helicase. Here, positive velocity means that its DNA is being unwound. Negative means that DNA is being rezipped. You can see that when Fred is high, you see a positive velocity. When Fred is low, you see a negative velocity. So I told you that our previous model was that this is the go signal, close conformation, and this is a no-go signal. But we had to actually revise the model again to say that this is actually a U-turn signal. So open conformation tells the enzyme to go back. So what's going on? The enzyme is unwinding in the closed form, following the 3' to 5' direction. And after it unwinds of maybe 10 base pairs or so, something happens to the enzyme to change its conformation to the open conformation, which is a U-turn signal. Then in this open conformation, the enzyme translocates on the other strand, also in the 3' to 5' direction. As it does that, DNA rezips in its wake, causing this DNA rezipping. After it fully rezips, DNA helicase switches to the other strand and repeats this many, many times. And structurally, we think that the two different structural conformations actually allow the enzyme to maintain contact with the duplex DNA using the 2B domain, and then swing over the rest of the domains from one strand to the other. So then this explains why RepX becomes a super helicase, because you are cross-linked into the closed form, and you cannot take the open form needed to strand switch. So it actually keeps going, cannot take a U-turn. So it's not like this RepX protein is a Porsche or Lamborghini. It's like a Honda Civic, okay? Honda Jazz or smaller, okay? But it's very persistent. It doesn't change direction, right? That's what makes it super. We have some data to suggest this idea that, well, basically unwind and rezip, just basically going back and forth like this is more like idling in a single position. It's essentially no-go signal. But in vivo, a partner protein comes to bind to stabilize the closed conformation. So you cannot take this U-turn, and then it keeps going in the same direction to make a helicase, a very powerful helicase. So we have data suggesting that for another protein, partner protein stabilizes the closed conformation. All right, so I'm coming to an end. We think that this monomeric super helicase without nuclease, it doesn't cut DNA, can be a very powerful tool for many biotechnological applications. One example is single molecule DNA sequencing using nanopores, because our enzyme can feed single-strand DNA into a pore for long distances. You can actually use it to sequence DNA of long lengths. Another possibility is to use the enzyme for isothermal DNA amplification. When you amplify DNA using a polymerase chain reaction, topic of a Nobel Prize too, you need to go up and down in temperature about 30 times to amplify the DNA, because to start the next cycle, you have to melt the duplex DNA to load the next primer. But if you have a super helicase that can do the unwinding for you, then you don't need to use an expensive machine to do this. You can just do it at a constant temperature. Then maybe you can use this to detect pathogenic DNA in Africa, for example, where you don't have access to expensive machines or even electricity. So let me show you two slides where we actually are making progress in these two different directions. So we are working with Jens Gunlach's lab in University of Washington, Seattle, to use our super helicase for nanopore DNA sequencing. The idea is that you have a nanopore in a membrane, and DNA is fed through the pore by a motor protein. So we provided our motor protein to them, and they were able to measure current fluctuations as a function of time. between many different levels, and actually this is about 2% of the entire length. You can just go on and on. Then you can discretize the data and then compare to predicted signal based on the known sequence dependence of the nanopore signal. You get a very good match between our data and then, so you're already actually getting sequence information on a really long, large length scale using the super helicase. The second application is isothermal DNA amplification. So here we are amplifying long DNA, 1 kilobase and 5 kilobase DNA, using our super helicase as an unwinding mechanism. Actually, others have done this similar things for short DNA, 100 base pair DNA, but now we can do this for much, much longer DNA. So what I'm showing you here is basically DNA amplification using DNA polymerase, primers, and template DNA. And if you use this wild-type protein without, actually this is a thermophilic version of the enzyme so that it functions at 60 degrees. If you use a wild-type, you get a smear in the gel. But if you use our super helicase, then at 3 different temperatures, you see nice 2 bands of amplification products. This is a kilobase pair product, and this is a 5 kilobase pair product. So you can actually amplify even very long DNA at a constant temperature. So this can be useful for, I think, many applications. All right, let me stop here. So my hero for the super helicase project is Sinan Aslan, who is now working for Illumina. And we work closely with Jan Shemna's group, first Tim and Matt Comstock, who was a joint postdoc. This is Tim Lohmann's lab, Haifeng Jia, who made the UVLD proteins, and Chris Thomas lead, who also provided us with some of the reagents. And this is the basic message of the paper. How to regulate enzymes function is by controlling its conformations, probably in vivo. You can use that information, then engineer biotechnologically useful super enzymes. Thank you.
So it's a beautiful talk. So I'm wondering for it's okay. I can yeah. For when you look at single molecules for when it is conformational change, when the conformational change when the molecule sample multiple conformation at a different speed range in your single molecule experiment, would you have sampling? Whether you will be able to sample all different conformations exchanging at a different speed.
So I assume that there are only 2 conformations when you analyze a single molecule data, but there may be more than 2. And in that case, then you'll have not just 2-step fluctuations, but you may have additional rates or additional different passages. And so whether we can detect more than 2 conformations, whether we can distinguish between different passages, will depend on our time and space resolution. So how different are the conformations in terms of flat values, and also how quickly are they changing? So yeah, so that's my answer.
My question is, this U-turn property is unique to this particular helicase, or it's quite common on the helicase?
So if I have to, okay, by the way, I lost my bet, right, 50 cents. And I told him that I will send him the money once the paper is published, but paper is published, we forgot. But anyways, I have to decide. So if I have to bet another 50 cents, okay. So this probably that behavior is common to many other enzymes. So people have reported repetitive unwinding of many different helicases, including my lab and many other labs. Basically, from a single molecule measurement, you can show that helicase can unwind some number of base pairs and it comes back and then basically goes back and forth. Most likely, you want to limit the extent of unwinding. when you only have the helicase itself until external signal comes in, partner protein or protein modification to be going in the same direction. So I think it is general. In fact, we are now trying to engineer RNA helicase, super helicases, because there are like probably more than 100 RNA helicases in our genome, specialized for different functions. If you can turn on the super helicase activity of a particular RNA helicase, you may be able to turn on a particular pathway inside a cell, right, on demand. So I think certain aspects of this must be general.
In this case, in your study here, what is the time resolution for on your x-axis is time, right? What is time resolution on that?
So, I have shown. So, let me first answer your question in terms of single molecule FRET measurements. Okay? So, if you push, you can go down to about a millisecond. In my lab, some other labs can do better if you use fancier technologies. But most of the data I showed today were obtained at around 50 millisecond time resolution. And the data I showed you where we combine FRET and optical tweezers, the FRET time resolution is also similar, but the noise is higher than usual because we only measure fluorescence signal about a third of the period. So we have signal is down by a factor of 3. Now, with optical tweezers, you also have down to about 10 millisecond time resolution for our instrument. But of course, it depends on the precision you're trying to achieve.
Humble average during a millisecond.
Exactly. So if you measure like high FRET for close conformation in 10 millisecond or 50 millisecond, it may still be an averaging of more than one state. So you'll have to go to higher time resolution to see this.
So what determines the speed? You show a lot of data which is almost parallel, but then there's also another transparency. It's not parallel.
Yes. So I think when I showed you just helicase moving on single-strand DNA, it was very uniform, right? But when I showed the data, show the data for super helicase unwinding, there were some variations, right? Maybe you're talking about that.
But there's another one. One of the transparency looks like there's several slopes of the position time.
Yeah. Let me try.
And nevertheless, you mentioned you said the speed is almost constant.
So when we measure unwinding by a super helicase in thousands of base pairs, we find that it actually, although it doesn't fall off, actually the enzyme does pause momentarily before unwinds a little bit, it pauses, unwinds a little bit, it pauses. And so depending on the pausing density for a particular trace, you may get a different impression of a different slope. Right? So that's maybe the one that you're talking about.
There's one which you show the almost constant of speed. The transparency before that. There's one particular one I can clearly see 3 slopes.
Oh. Yeah, maybe you can discuss it a little later. Yeah.
So I have a generic question. I mean, to what degree all this nice single molecular experiment result can be applied to a live cell, where in the cell you have many other things in the thing. We know that you can just the knowledge we learn from single molecular can be 100% applied to a single cell real action, or there's some modification? Because in the cell, okay, so there are many other activities. It's a soup, so it's not single molecule anymore.
This is a $55 million question because I need that much money. So obviously, all the data I have shown you today were single molecule reactions in isolation. So these proteins do not function in isolation inside a cell, right? So, we actually started a new center in Illinois called for Center for the Physics of Living Cells. I was the director of the center for the first 7 years, and the vision was to really make the measurement more relevant to what's really going inside a cell. And one way of doing this is to perform some of the measurements inside a living cell, which is not so easy, but in many cases possible these days. I'm sure many of you are working on these issues. Another direction to pursue is to go extreme in vitro. So the limitation of the studies we are doing is that you only have maybe 1 or 2 different kinds of proteins on a DNA. But to go through entire process DNA replication, you need many, many more proteins. So then if you have multicolor fluorescence and maybe multi-axis optical tweezers, you may be able to actually measure even more complex systems with more components. So we also made that progress in that direction. And so this is actually ultra-resolution tweezers plus fluorescence is one example of this kind. Instead of just measuring one number as a function of time, we are measuring 2 different numbers. We can push it to 3, 4, 5 different numbers. Actually, there's no issue with the measurement itself. The difficulty is actually the labeling of these proteins, different colors. That becomes a remaining challenge. So,
up to now, do we have an example that single molecule to get a consistent result inside the cell, maybe one particular one, or do we have a counter examples that what we learn in single molecule experiment does not apply to a live cell? I just want to know, do we know anything now?
So people, let me give you one example where people have used single molecule imaging, even optical tweezers, to measure motor protein functions inside the living cell. And actually, you don't see exactly the same thing between in vitro and in vivo, but in many cases, in vitro measurements, single molecule measurement, can inform the interpretation of the in vivo data. Right? So, ideally, you want to perform both whenever possible. And I think there are many examples where in vitro measurements does not hold true in vivo, or vice versa. There are also things called in vivo artifacts, right? You make a conclusion based on in vivo data, but you neglect the fact that actually in vivo there are some other things you didn't know about. So you may draw a conclusion about a particular molecule based on in vivo data. That may be wrong because there are hidden entities inside the cell that complicate your interpretation. So you have to go, back to back together. So it's a challenge that we have to think about all the time, right? So when in vitro versus in vivo, right? There's always tension between the two, and if you're under tension, actually, then that your research can be kept honest and healthy. So,
thank you.
Good question. Yes.
I have a small question.
Yeah.
I'm just wondering whether the sequence of the DNA sequence will interfere the pattern or movement of the helicase. That's a small question.
Yeah. In our experiment of the super helicase, we haven't studied the sequence dependence yet, and my prediction would be that it will be depend on the stability of the DNA speed, but I don't know for sure. For other helicases where people measure very limited amount of unwinding before coming back, there how far the helicase can go before it comes back depends on the DNA stability. If you have many GC base pairs, it goes maybe 5 base pairs, it comes back. If you have many 80 base pairs, it can go 15 base pairs. So there is actually balance between the stability of the enzyme, DNA, and how far the enzyme can go.
Question? Yeah, I have one question.
Okay.
So, DNA helicase is a lot in in cell, right? So, and there are a lot of, I'll say, diverse DNA helicase. So, sometimes when you show the two, how say, competing models, so is it possible that one model, how say, fit with a certain, how say, part, certain portions of the DNA helicase, and the other model fit with, how say, the other part of the DNA helicase?
Yeah, that's a very good point, and that's the reason we actually, in our project, we studied all three proteins in the same family. They're not exactly the same. They function in slightly different processes, but we find them to be essentially identical in their behavior in this context. Oh, by the way, one thing that we are doing right now, we can actually have a helicase mutation that can stabilize a closed form, so to mimic the super helicase, but not quite as super. And then we are doing some in vivo measurements of the helicase function in a particular process using genetic experiments to compare our in vitro data and in vivo data. And we see actually pretty good correspondence that gives you confidence that something is things are actually reasonably working in the manner that we proposed. So I think that one nice way of comparing in vitro in vivo is that to make mutations to change single molecule behavior in vitro and see what the consequence is for in vivo observations.
And let's thank the speaker again.